Ya kok wifi. Tes. Udah nggak dua ya? Dini, halo Din. Dengar suara saya nggak Din? Dengar ya? Jelas?
Iya, udah bisa Mbak. Oke, jelas.
Oke, thank you Din.
Mohon maaf gak bisa hadir di sana.
Iya. Di kantor ini. Oke, kemudian ini ada Pak Lili, selamat datang Pak Lili. Ada Ririn. Selamat pagi Bapak dan Ibu sekalian serta adik-adik sekalian. Karena waktunya sudah saatnya dimulai, jadi langsung kita mulai. Ini juga selamat datang untuk Bapak Ibu serta adik-adik mahasiswa yang hadir secara online. Kita akan segera memulai acaranya. I'm sorry, I will start the lecture. Okay. Ladies and gentlemen, and also for all the student, master, doctoral, and also undergraduate student, because of the time, we will start this general lecture soon. Assalamualaikum warahmatullahi wabarakatuh. Welcome to Professor Futoshi Ishiguri to Indonesia and also to our faculty. We are very pleased that you can visit our faculty in this occasion to give a general lecture. Welcome also to Ph.D. student and undergraduate of Professor Ishiguri Sensei. And good morning for all the lecturers, master and doctoral student, and also for undergraduate student. Welcome also to the ladies and gentlemen and the student who attend this meeting online. This general lecture is held by offline and also online. Thank you very much for your presence in this activity. Well, all right, ladies and gentlemen, before starting the general lecture, I will read a brief curriculum vitae of the Professor Futoshi Ishiguri. Professor Futoshi Ishiguri now is in academic appointment is associate professor in the Faculty of Agriculture, Utsunomiya University, and his research interest is in wood anatomy and wood properties, especially for relationship between growth and wood property in plantation trees, and then anatomy and lignin distribution of reaction within angiosperm, and then the changes of the physical and chemical properties of wood by heat treatment. And for educational background, Professor Ishiguri bachelor degree from Utsunomiya University and also for the master degree. For the Ph.D. degree is from Tokyo University of Agriculture and Technology. And Professor Ishiguri already have some fellowship and also awards. And for the publication, Professor Ishiguri have so many publication, international publication, more 100. So I could not read one by one because so many publication from Professor Futoshi Ishiguri. Well, as we know, related to the title of this general lecture, tree breeding is an important aspect in the silvicultural practice. In the past, especially in Indonesia, breeding was still focused on tree growth. In the last few years, we have started involving aspect in wood quality in tree breeding activities in Indonesia with collaboration with Professor Ishiguri. We have to realize that wood quality is an important factor and cannot be separated from tree breeding activities. Maybe next we immediately invite Professor Futoshi Ishiguri to give a lecture entitled Tree Breeding for Wood Quality. Professor Ishiguri, time is yours.
Thank you, everyone. Thank you very much. So actually, that every year I come share past about 10 years we can. It's my pleasure to okay. Anyway, I today I would like to talk about myself. Okay. Okay. Let me introduce myself. Dr. Fanny already introduced me. My name is Futoshi Ishiguri. Usually the people called me the Ishiguri sensei. My research interest is wood anatomy and wood quality, and I joined the several societies, some of them international society and the domestic society. And key word is the wood. I work in the wood anatomy and wood quality, so the society also treat with the wood. Okay. And my research topics is simply divided into the 4 topics. The one is relationships between growth characteristics and wood properties in tropical species. And the second one, the tree breeding for wood quality in Japanese softwood species. The third one is anatomical and chemical characteristics related to pulp and paper quality. And the final one is the relationships among standing tree characteristics, log quality, lumber properties of trees. And the below 4 papers is recent publications in from my laboratory.
Recording in progress.
Almost all authors are my PhD student. Okay. Okay. So when I come in here and to make some lecture, always I use this slide. Many people ask me about the first one is this first time to come to here? Sorry, not this is fast. And I come so many times. And the next one is the how many times. I'm sorry, I forget. Over 20 times maybe. At 2002, I come in here to join the PRWC meeting. So I maybe I met with the Professor Ganis. Maybe since just visit Yamagata before, I remember. At the time, just the student. And some people about where did you go in Indonesia. I visited so many places to collect some samples. And final one is the Bisa Bicara Bahasa Indonesia. Tidak bisa. Okay. So now we move to the next topics about the. I'm come from the Utsunomiya University, so I have to explain about my university. First of all, I would like to explain about the where is Utsunomiya. Actually, the Utsunomiya is very near from the capital city, Tokyo. It's about 100 kilometers to the north side, and it takes just within 1 hour by the Shinkansen Super Express train. This picture. This is a Utsunomiya station, and about 1 hour by this train. And my university was started in 1949 after the World War II. As one of the national university, and the university, especially for the School of Agriculture, based on the previous Utsunomiya Higher Agriculture and Forestry College established in the 1922. So the School of Agriculture last year 100 years old, 100-year-old. And of course, during the COVID-19, so we don't have any celebration ceremony. But this year, we will make some celebration ceremony. And now that we have the 5 faculties, regional design, international study, and engineering. Compared to your university, my university is very small. And among the national university in Japan, my university is very small. And you see the several photographs. The color picture is the present campus. This middle part of the red color flowers. That is, I took photo in this May. And you see the beautiful sakura in April. Okay. And I would talk about my laboratory, the origin of my laboratory. As I told before, the establishment of the college at 1922, and the laboratory itself established in the 1930s. At that time, the name is the Laboratory of Forest Utilization. So, including not only the forest products, but also the forest engineering issues, the logging activities, and some others. And after World War II, 1947, the laboratory independent from the forest utilization, and we established the Laboratory of Forest Products. And the photographs. The middle one is the most oldest microtome. Maybe we bought it around the 1930s, almost 100 years ago. And you see the preparat. This is a bamboo, and according to the information in here, maybe this one produced in the 1933, and the first professor, he made it and still keep this one. And he summarized the many data in his book. And like this slide, he took this slide and make such kind of the photographs. And he also focused on the strength property. So you see here the testing machine about 100 years ago. So now our laboratory focus on one is wood anatomy and wood quality, and the other is the forest chemistry. And I'm work in the wood anatomy and wood quality, and my professor at Shinso Yokota focus on the forest chemistry. Okay, and I list up the Indonesian PhDs from my laboratory. Dr. Fanny, 2012. Yesterday we talked about almost 10 years or more past, so I was surprised. And next, today's moderator, Dr. Fanny. And so many students, unfortunately, so many students come and get PhD. And now below part, two persons. One is the Parensis in the 2003. She already finished the defense, and maybe she will get the PhD. And the final one is just last year come Masendra-san, and maybe he will get to the PhD in 2025, hopefully. Okay, so my laboratory the huge relationship with Indonesia. And I collected samples not so many countries, but several Asian countries, not only the tropical country, the Indonesia, Malaysia, and also Thailand, but also the very cold climate in the Mongolia. I listed up the sample I ever collected in here. There's so many samples, especially for the Indonesia that we collected so many samples. Mainly the fast-growing tree species. And when we collect samples in the field, I list the example, 3 examples. The one is already 10 years or more past, and this one 10 years past about in the Thailand, and this one most latest, but before COVID-19 in Malaysia. We collected samples in the field, and not only the research activities, but also we sometimes go to the shopping, sightseeing, and dinner, and I think that one is set. Research and some enjoying something that is set. And after back to my country, we analyze the samples, and finally we published some result, like these papers. Okay. So now we move to the main topics about the tree breeding for wood quality. We would like to introduce the first wood quality itself, and the second part I would like to talk about the tree breeding for wood quality. Today I talk about the very, very simple and basic things only. And after my presentation, my PhD student will show some recent results that is much academic. For me, just like the undergraduate class. So first of all, I have to apologize for you. Okay. So now I talk about the introduction of wood quality. So what is a wood quality? So I list up the definition of the wood qualities. The upper part, sometimes the people consider that it's not wood quality. For example, the stem shape, branch, growth link structure. Especially for the stem shape and branch, the people sometimes feel strange. But the wood quality means the quality means sometimes we use the wood property, word of the wood properties, and sometimes we use the word of wood quality, property and quality. Sometimes the people said it's same, but in my slide I use the different word. The property itself, just property. And quality means that if we already decide some final product, for example, if we want to make the structural lumber needs the strength properties, the dimensional stabilities. So such kind of factor, if it's good, we call it wood quality is good. So I use I separately use the quality and property. So that's why the stem shape and branches sometimes affect on the final product. So that's why I included in the stem shape and branches and the wood quality. And first 3 parts, maybe the growth traits, closely related to the growth, not wood itself, the growth. And the bottom part, number 4 to the 9, that is closely related to the wood itself. And for example, the number 4, basic density, wood density, and number 5, fiber orientation. 6, the length of the truncated or the fiber length, cell length, that is closely related to the pulp and paper qualities. And also the number 7, also the cell morphologies. This is also very important for the pulp and paper quality. And also the wood is consisted of the cells, organ cells. So the cell morphology is quite important for the every final product qualities. And if we want to use the wood as structural lumber, the microfiber angle is very important. And the final part, the number 9 is the reaction wood the abnormal xylem tissue, we feel that if we use the wood, this is abnormal. But it's the reaction wood itself, the tree need to alive. If the stem bent, the stem try to become straight. So at that time the reaction wood formed. So tree need reaction wood, but human beings don't need. So this is contradict things, but important issue as a wood quality. And as I told you before, the desirable wood quality is quite old word. And the desirable wood quality depends on the quality of the final product. Because as I told before, if we want to get the structural lumber, maybe the higher wood density and lower microfiber angled wood is very important. But if we want to make the pulp and paper products, maybe the strength properties don't need higher strength properties, don't need to for the pulp and paper. For the charcoal or the firewood, we don't need to the lower microfiber angles. It doesn't matter microfiber angle. So we have to consider the final products, and then the which factor, which traits, which properties is needed for that product. Then we have to consider such kind of things. So that's why some wood, the people just easily ask me, is it good or not? Good wood, good tree, good wood. But it's very difficult to answer. Because this is good for the structural lumber, but this is not good for the pulp and paper products. Something like that. So we have to consider the desirable wood qualities depends on the quality of the final product. Wood density is very important with trait. Already, as you know, the wood density is closely related to the shrinkage values and the strength properties. We can easily estimate the strength properties by using the densities. So density is very important. And density, it's not stable from the piece to bark and not stable among the trees. Sometimes you see some large variations. The density values are varied from the piece to bark. This is example from the Japanese softwood species. Maybe you don't know the scientific name, but maybe you know the Pinus species. Your country has the Pinus melkshi, so you know Pinus. It's very similar species. And the Pinus species show the density show the lower value in the piece side and gradually increase about the bark. But the Japanese cedar, the typical Japanese plantation species, this is not. Most inner part, the piece size shows sometimes the higher value of the density, and gradually decrease, and then again the increase, something like that. So the radial variation profile depends on the species. So if we want to effectively utilize the wood, we need to clarify such kind of the radial variations. Okay, and also the within tree variation. But usually within tree variation, if we collect samples at breast height position around the 1.3 meter height, the value is also similar. We can speculate if we know the breast height positions data, maybe we can speculate the whole trees. And the next is the trachate length, the cell length. In your country, softwood species is very rare, so almost hardwood species. So that's why I should say the wood fiber length that is much popular in here. But anyway, the trachate length, we know the very old law. We call it Sanyo's law. It describes about the length length of the trachea shorter in the most shortest in the peace position, and gradually increase, and then the become stable, and start to stable. That is around the 20 annual rings. And here the annual ring also not so popular, but in temperate zone the growth season is just one time in 1 year, so we find the growth rings. So that's why we determine such kind of one by using the growth rings that can be arranged. So anyway, the most shortest, and then increase and become stable. So I showed in here. Again, the Japanese cedar and the pinus, the Japanese red pine, and even in the stable value, here is about 3 millimeter, and here is maybe the 4 or 5 millimeter, a little bit different, but the shape, the increase and become stable, that is the same in the both species. And the start to stabilize, that is the both the same in the around 50 to the 20 annual length from the piece. That is the same. So the value itself sometimes different, but the same trend. And next is the microfibril angle. This is also very important. The cell layers, and the most thick layer is S2 layer, and we determine these layers microfibril angles. And the microfibril angles, usually the pitch side show the higher value and dramatically decrease, and then become stable around the 10 degree or less than the 10 degree. And the microfibril angle is quite important for the wood light. For example, the MFA related to the Young's modulus, lower value of the MFA wood with the lower value of MFA show the higher value of the Young's modulus, and also closely related to the shrinkage value, the stability of the wood. If the lower value of the microfibril angle, maybe the shrinkage value is quite low. So the compression wood, sometimes the microfibril angle over the 40 degree, so the longitudinal shrinkage is very high in the compression wood. So the microfibril angle is very important. And I would like to introduce about the concept of the juvenile and mature wood. This is come from the softwood species. So in case of the softwood species, juvenile and mature wood concept is very easy. But in your country, almost all one is the hardwood species. So hardwood species, sometimes if you Googling, maybe you find this word even in the hardwood species. But actually, the hardwood species, this concept is sometimes okay, but sometimes not okay. So that still difficult to use this word. This is my opinion. The many researchers try to research about this matter. So some of them use this word, some of them not use. Me not use. So up to you. Okay. Anyway, we sometimes determine the juvenile wood and the mature wood by using the length of the cell. In this case, the trachea length and also the microfibril angle of radial variation of these factors. And the microfibril angles decrease and become stable. And the trachea lengths increase and then become stable. So starting point of becoming stable, that is as I told you before, around the 20 annual lengths from the piece. We call it the Campbell age, 20 years Campbell ages. So in general, here is we call it juvenile wood, and here we call it mature wood. And in the stem, at one stem, the juvenile wood exists from the bottom to the up. And the upper part, all one is the juvenile wood. And the lower part, maybe the elder tree has the juvenile portion in the inside of the trees. And outside, maybe we find some the mature wood. And in the logs, in the inside juvenile wood, it's unstable wood quality. And the outside usually mature wood, so lower microfibrillae angle and the longer fibers. So if we collect some lumbers like these pictures, the middle portion may be including the juvenile wood and outer part including the mature wood. So what does it mean? It means if we collect the structural lumber need the strength properties. So if we collect the lumber from the middle portion, that is consists of the juvenile wood. So it shows the lower Young's modulus, not so good dimensional stabilities. But if we collect even in the same wood, one log, if we collect the lumber from the outside to consist of the mature wood, maybe this portion this lumber show the higher Young's modulus than these lumbers. So even in a tree, a log, quite different. So we have to consider such the juvenile wood and mature wood concept. We have to consider it. And also this is very important things. Juvenile wood does not change to the mature wood. Sometimes in here you may not drink the wine, but wine, whiskey, some alcohol, keep the several years, 10 years, 20 years, and then it become matured and it become expensive. Taste, I don't know exactly. I never drink the expensive ones, so I don't know exactly. But people said it become the mature. But in this case, not. Keep in here, juvenile is juvenile forever. So not change, okay. Okay. So this is example of the juvenile wood and mature wood differences. And anyway, the juvenile wood showed the lower Young's modulus. And shrinkage, I'm sorry, this is wrong. The opposite. The juvenile wood showed the higher shrinkage values. So if we use the wood as a solid lumber, it's better to use the mature wood. And next topic is how to determine the boundary between the juvenile wood and mature wood. Of course, we cannot determine by the naked eye if we say just look the wood and this is mature, this is juvenile. That is quite difficult. So what should we do? Usually the very old literature. When I was student, not so old in the 1981, but still the 2023. So the 40 years passed, maybe about 40 years passed. The Professor Shiokura published one famous paper to determine the boundary between the juvenile wood by using the trachite lengths. And he measured the trachite length in the first annual length first, and then the next is the several intervals, for example, the 3 years intervals or the 5 years intervals. And this one is not first and 3, but anyway, the circular shape, that is the actual data of the trachite lengths. And next step is he tried to determine the formula by using the actual values. The formula is usually the logarithmic formula. Y is trachite length, and ln X is the Campbell age or the annual ring number, and then plus B. That is a simple regression formula. And he obtained this formula. He calculated the each, between the measured one. As I told people that we measured 2 or 3 or 5 years of intervals, but by using this formula, this regression formula, we can calculate each years how the 1 year increase how many trachyte lengths. So after estimate every year's length, trachyte length, and then calculating the increase ratio per year, 1 year, how increase the trachyte length. And he considered the determining of the boundary between the juvenile wood and the mature wood is quite good adapt with the increase ratio less than 1%. So we usually use this method, and this is example. This one is increase ratio. The increase ratio gradually decrease, and then the less than 1% around the between the 10 to the 20 annual length. So this is the model. So model quite adapted with the actual phenomena. And also we can determine the boundaries by using another method, using the microfibular angle. And now that we try to introduce some mathematical modeling using some statistical method. So for the statistical method, first of all, we try to modeling about the radial variation. In this case, the radial variation of the trachyte length, and also we also try to modeling about the radial variation of some other wood types. And in case of the modeling, now that I show you the 3 simple models. Linear, logarithmic, and quadratic functions. And sometimes we also use the different models, like the Michaelis-Menten formula. That is usually the chemistry use the enzymatic reactions. We also use such kind of the formula. And now that we try to use the mixed effect models, and the mixed effect models goes to for example, the slope or intercept, and usually in this case the tree, we see the trees, word of the trees in here. This is random effect. So random effect of the individual trees, maybe the value itself, buried in the individual trees. So how differed we put these this effect in here and here, and finally we select which one is most adapted, and then use the explaining about the radial variation. By using the model, and then calculate the changing ratio of each properties. And after that, the changing ratio gradually decreases and becomes stable. So if the values become stable, of course the changing ratio also becomes stable in the below lower values. And after that, we try to find the intersection point of this value, and then decide the boundary between the juvenile wood and the mature wood. This research result, maybe the next speaker, my PhD student, will explain. And if you need the Indonesian explanation, Dr. Agus will explain. Okay? Okay. Okay, so now that we try to determine the boundary like this. And strength property itself very important. So you already know the elasticity, plasticity, stress, strain. Maybe you already know, so that I skip this slide. And for the strength properties, usually the destructive test is mainly conducted, but sometimes the destructive test is not so good. So we need to know that some strength property non-destructively. So this is one of the typical example of the non-destructive test for obtaining the Young's modulus, that we call it dynamic Young's modulus. And this is log, and tapped by the small hammers of the one cross section, and the other section we put the microphone, and the data goes to the fast Fourier transform analyzer. Now the very good I don't know how to say that this machine itself very expensive previously. Now you can download the free software application, and sometimes the data looks like strange, but mainly okay. So the estimation that is good. So just you can calculate the Young's modulus by using your smartphone. That is very good. And anyway, to get some fast resonance frequency by using the FFT analyzer, and after that we use this formula and get the Young's modulus. And this Young's modulus value itself closely related to the destructive test result, like the MOE and MOR. So very deep and good method. And also, Dr. Fanny also focused on this study, to be sometimes measure the stress wave velocity of the trees. This is also the very simple and fast method, and she just put these sensors, and the wave goes to the another sensor, and this one is just a timer. So how many minutes, not minutes, maybe the microsecond, not minutes. If we know the time, we can calculate the velocity, the speed. So by using that speed, we speculate the mechanical properties. This value is also quite how to say, the high correlation coefficient between this value and destructive MOE and MOR. So this is very good, especially for the tree bleeding matter. The people use this method. And also we need finally the destructive test. And destructive test by using the UTM. In my laboratory, we have the three testing machine, the 5,000 Newton, 200 Newton, and 50 kilonewton. And sometimes we focus on this full-size specimen. At that time, we collaborate with the prefecture, local governmental forestry center, and we tested this test, bending test. And also, we sometimes cannot collect the trees. We don't get any permission to cut. At that time, sometimes we collect the very, very small, looks like the straw shape, the samples. And by using that straw shape samples, just diameter 5 millimeter, using this machine, we can estimate the compressive strength. The data is showed. Data is the calibration between the normal small clear specimen and the very small samples. Okay, by using such kind of the equipment, we collect the strength properties data. Okay, so now let's move to the tree breeding for wood quality, the next topics, the main topics. Before starting about wood, I would like to introduce the potato. You know the potato. And in Japan, I collected this photo from the governmental Ministry of the Agriculture, next is Forestry and Fishery. And they explain about so many potatoes. And when I was a child, only this one. We call it danshaku, this one. And now so many potatoes, and some of them taste is good for the fried potato, French fry, and some of them should use for the korokke. It's not in English, but korokke. So many potatoes, and you see the different shapes, and the color sometimes also the different, but the scientific name is the same. Only one scientific name, but it means the same species, but morphology is quite different, and taste also quite different. So someone tried to make the new potato for some specific food, something like that. I don't want to explain about potato. Actually, I want to explain about wood. So in this case, the taste of the potato is quite similar with the property of the wood, because the taste, that is the final product of the vegetables, and wood properties, that is the final products need. So wood property in tree are just like taste of the potato. So as you know, so this slide showed wood property, the potato's taste is controlled by the genetics. Its cultivar differences makes such kind of the morphology differences or the taste differences. So wood properties differences also come from the genetics. So this slide here, you see the column, the transverse section of the columns. This is all one is the same species, but the different genetics, genotypes. So this one, after drying, not deform, but this one quite showed a huge twist. So if we make some good clear lumber, maybe we have to cut again, and then the sectional size it becomes small. So almost all one is that become waste. The same species, which one is better? Maybe we should take this one. So this is come from the genetics. Of course, not only the genetics, environmental factors also affect on this phenomena, but mainly the genetics. So that's why we conduct the tree breeding. And the typical tree breeding, we try to find some good shaped trees, faster growing trees in the plantations or the natural forest, and collect some seed or the triggers and plant in some places and check the conditions again and select this more good samples, good mother trees or some good genetic trees. And we try to conduct the several times, so many times, and then finally get the good wood, maybe. and select plant, select plant, select plant, select, and select plant, select plant, so many times. The one generation select plant, and then the next generation, second generation, third generation, fourth generation. Now the Indonesian acacia mangium, you know the acacia mangium, fast growing tree species, industrial use, that is already goes to the third generation. In Japan, Japanese cedar, second generation, already passing through. Your countries, the tree grow well, so that's why they're very fast, very, very fast. In our case, it takes long time, so need long time. So that's why some people try to reduce the breeding time, of course, by using the molecular biological techniques, by using such kind of techniques, DNA and some others, and then try to reduce the time. So maybe in your country, the one generation is within 10 years. So if we reduce much more shorter, it's very attractive. Okay. So what kind of the important traits for how to select the superior trees? What is the superior? Sometimes we find so many traits: radial growth, height growth. Radial growth, height growth, tree form, so many things. And radial growth and height growth is very easy to consider. Fast growing, bigger size, it looks like good. It's true, okay. And how about tree form? Tree form, maybe the straight one is much better to use the solid lumber production. If just the pulp and paper products, maybe not. But sometimes need the straight one. Okay. But this is growth light. And growth light also need the others, like the resistance for the disease and resistance for the insect. That is very important. And also this is also very important, resistance for the environmental factors. In Japan, for example, in the winter season become very cold, and some places has much snow. Some snow damages, so is to the snow, or sometimes in the summer season typhoon attacks, very huge window below, and then to avoid such kind of things, we need some resistances. Such kind of the growth light. This is not grow well. Very good, but grow well. Should plant the good growth, resistance for the disease, and so first priority, primary objective is growth lights. Sorry. Next is the wood lights. So better to select the 1 times 2 characteristics, 3 characteristics. That is. We put the growth light in the x-axis and y-axis. Negative correlation with the growth light and the wood light. Case, the characteristics makes the lower wood properties, lower wood qualities. Get the good one at once, but can collect, for example, this one is good for both faster growing and good. No correlation. If we cannot find any correlations, it means endoxide. But we can collect. For example, this one select, this one select, this one select. And then maybe the both change. So such we have the clone BC, 3 types of the clones, and we planted the 3 trees in each clone and measured the stem diameter, and then the mean value is A, B, C, 10.5, 12.8, and 15.9 respectively. In that case, how the diameter growth characteristics goes to the next generation. We make the simple linear models like this one, the mu plus clone plus E. The E is residual. In this case, they mainly come from the environmental issues. And after that, by using the variance of the clone and the residuals, we can calculate the repeatabilities. Sometimes we call it the heritability or the repeatabilities. And in this case, the repeatability is 0.9 something, so very high values. It means this stem diameter is right, maybe the highly inheritance to the next generation. This is, you can calculate by using the simple ANOVA test. Maybe the student study in the statistical class, maybe please remember the one-way analysis, two-way analysis. Maybe you can calculate by yourself. And in this case, we use the mixed effect model, so the little bit different systems, but the scheme is similar. And then also this is sometimes the progeny test to use the different places, the same genetic set, but we planted the site 1, site 2, site 3, the different places. The environmental condition is quite different, 3 places. And then after that, we can estimate the repeatability too. So in this case, the repeatability is the 0.27, about 30% diameter traits heritable in the next generation, something like that. So we calculate these values to clarify how this trait goes to the next generation. And early selection is also a very important idea. This is the 5 years old Japanese seed, and 5 years go old, and after 15 years they become 20 years old, like this shape, and 30 years like this shape. And in the case of the Japanese seed, at least maybe the 40 years needed for harvesting. So the next 10 years we still need to wait. But anyway, the 5 years, 20 years, 30 years we have, for example. And then some traits, 5 years data and 20 years data or the 30 years data, if we find the highly positive correlations, or the negative correlation is also okay, the significant correlation if we find, in that case we can estimate by using the 5 years old values to the unknown future values we can estimate. So in the growth rate, we have to wait. If we make this graph, in case of the growth rate, we have to wait because the 5 years, and if we get the 20 years old, we have to wait for the 20 years. So need time, but we are lucky. We focus on the wood. So that's why lucky? Because the wood formed in here, maybe the 5 years old. Age 5 years old makes this wood. And at the 20 years, maybe like this portion maybe made. And 30 years made in here. So at once, if we have the elder trees, at once we can calculate these phenomena. And if we find the highly positive or negative correlation between the younger age and elder age, we can, if we find like this, we can select the good trees at the 5 years old. So wood, if we find some elder trees, very easy to say something. So this is the final topics that I just briefly explain about the case study in the Eucalyptus camaldulensis in Thailand. I used some Indonesian study. I'm sorry, I don't have enough time, so I used the eucalyptus study in Thailand. But the concept is the same. Okay. So in the Eucalyptus camaldulensis in Thailand, they tried to use making the pulp and paper, and they collected 120 families in this at the progeny test site. And we collected some samples, the core samples and some data in standing trees. Among them, the 50 families, the 78 trees, and 12 years old, we collected some data. And this is the result of this diameter, height, and stress wave velocity that is quite closely related to the Young's modulus, and release surface strain. Maybe you remember this word. Professor Nugroho teaching about the release surface strain. So compared to my student, maybe the UGM student much familiar with this word, I think. So in this case, I applied the simple ANOVA test, so that we got the F value. If this value higher, we have some possibility to, we find among family variations, family differed among some traits differed among the families. In this case, the stem diameter, tree height, and surface wave velocity showed significant F values. So it means the family with the superior growth rate and the high Young's modulus we can select. And in this case, as I forget to explain, this progeny test site already the second generation. So already first generation already selected based on the growth characteristics. So already first generation showed good growth characteristics compared to the conventional seedlings. And even in the second, already one selected, and already selected one we again select, but still we have the possibility to select the good growth characteristics. And if you see the F value if we compare the F value, here the 2 point something, 2 point something, and here the 3 point something. It means the first generation not selected based on the Young's modulus. The second generation also not based on the Young's modulus. But growth characteristics already selected. So that's why the diversity becomes smaller than the Young's modulus. So we can check these values by using just the simple ANOVA test. And unfortunately, the least surface strains cannot find any significant differences. So maybe we try to find some reasons. One is the variation of the among the families not so large. So this, but I think that this one maybe the closely related to the genetics. So maybe we find some reasons, but still now we don't know why it not showed no significant. Okay, and same things goes to the wood properties. The almost all wood property, especially for the core and outer. Core and outer means the near the piece and the near the bark side. Outer means the bark side. Especially for the bark side, almost all wood characteristics. I'm sorry, this is CM, compressive strength. The almost all characteristics show the significant among family variations. So it means especially for the core wood, maybe we can improve by using the tree breeding techniques. And also this is the result of the correlation between the diameter and the other traits. And diameter and the tree height show the high correlation, positive correlation, means the bigger size tree may be the dingy tree. And how about the diameter and stress velocity? Youngest modulus and diameter, radial growth rate is not related. So it means that the other one is also the same. So it means the wood trait are mainly basically independent from the growth rates. So it suggests that the families with superior characteristics of the both growth and wood trait can be selected from even in the second generation. And this slide shows the correlation between the core and outer core and outer. If we find the positive with the negative correlation, significant correlation, we can apply the early selections. Unfortunately, in this case, only the cell lengths can find the positive correlation. So in this case, the cell lengths, if we need the longer fiber for the pulp and paper, in the small tree, examine the wood fiber lengths, and if that will show the longer fiber, maybe 10 years later, we also get the longer fibers. So we can apply the early selection in this traits. But unfortunately, the other one, usually we find the basic density, but unfortunately, in this case, we cannot find any significant correlations. I don't know reasons, but we should try to get much more data. Okay. Okay, this is final slide from my presentation. The significant among family differences were recognized in some traits. So it means a selection with superior family is possible. And no significant correlations were found between growth traits and wood traits. The wood traits are independent from good traits. Sometimes we cannot find this one. So at that time we have to discuss how should we do. And superior family with both growth and wood traits is possible. And these facts strongly support that in case of the eucalyptus in Thailand, we can convert the utilization method from the pulp and paper products to the solid wood production. As you know, the solid wood production is has an economic value compared to the pulp and paper products. Pulp and paper product itself, that is also very good, very good. But sometimes if we need much money, sometimes the good tree should go to the solid wood production. And not so good, for example, the shape and some other not so good characteristics, it can use the pulp and paper products. Okay, that's all my presentation. Thank you very much for your kind attention.
Thank you very much to Professor Ishiguri Sensei, which was a very interesting and useful presentation for all of us, especially in adding the knowledge in the aspect of the wood quality in relation to the breeding activity. Arne Zuzang also will do some. After this is question. Okay. Furthermore, everyone are welcoming if you have any question. Okay. First,
Okay. My name is Rafif. I'm a master student. I'm interested with the explanation between you mentioned the positive correlation. It is possible to select three. It is good, but you mentioned that if negative correlation in the selection is difficult. But what if the trait we want is to lower the trait? Like if, for example, if we want lignin content to be lower, but so is that will be good?
Okay, thank you very much for your question. Yes, I skip such kind of things. Sometimes the lower value is good, sometimes higher value is good. So the negative, positive is a bit difficult to explain. That is true. Yes, the lignin content for the pulp and paper products, maybe the lower one is much better. So in that case, negative is good. Yes. Simply like this, right? So, of course, the exceptional things we find, of course, yes.
I'm interested with the earlier slides. You showed us many trees from many regions that you have observed. You make the color difference, and I recognize some of it's a softwood and some of hardwood. But what makes the difference between one color to each other? Maybe what's interesting about that?
You looks like very clever. I'm sorry, this is used for the different the symposium in Japan, and at that time I explained about the fast-growing tree species. So red color shows it looks like the fast-growing tree species, and so that's why your countries has so many fast-growing tree species, and in Japan not so much. And the gray color, that time they just only focus fast-growing tree species, so that's why the gray color ignored. So ignored species, and green color shows the border being ignored and the fast-growing, exactly fast-growing, and definition of the fast-growing tree species difficult. Country maybe the 4 years old, 5 years old, maybe the people consider fast growing, but in Japan 4, 5, maybe impossible. 10, 20 looks like the. That conference I explained it. Korea, very cold, fast growing. Cannot wait 30 years. But so the part of the fast growing is very different. Anyway, this is the fast growing tree species.
Dr. Ganis Lukmandaru, please.
This is very interesting for the presentation. This is the simple question. So about the modeling, how many samples in radial variation which we need to get good models? Yes, yes, on your experience. The second one is what is the target of coefficient determination or R square, the minimum? Okay, thank you.
Okay. Very, very good question for us. If we collected so many large number of the samples, the accuracy of the model increase. And the shield scan, sometimes X-ray techniques. So other case 1,000, 2,000, and maybe that case, I guess the company make donation. So they try to explain simply explain how to make the good products. The company made some samples or I don't know exactly the money or not. So
Expensive.
yeah. But in Japan, of course, not so much. But the diameter and some close right, usually that one is the non-destructive. So the researcher used a huge number of the samples, like in the Forestry Breeding Center used a huge number of the samples. But in this case, the minimum 3, but the 3 sometimes makes conflict. And the second question is the R square of something. After introducing the modeling group techniques, we not use not so much the R square and some others. How model adapt? That one is very important. So, and of course we can calculate the R square values and some others, but how we can explain these phenomena by using the models. And the model gives some, if we conduct the modeling, we get some other variance dated and some other parameters. Model itself and mixed effect model has, we got the 2 parameters, the fixed effect parameters and the random effect parameters. And by using the value itself, the modeling value itself, parameter value itself, we consider something. Not use, yeah. No, do not use.
We would like to in the Zoom, please raise hand. Dr. Hairi.
Thank you, Ishiguri Sensei, for your nice and comprehensive presentation. So, in this day, many countries start to implement counting the carbon storage or carbon trading. Maybe you know about that. So, my question is, what do you think the wood parameter or wood trades that important for this purpose? Do you have any opinion about that?
You mean the carbon sink?
Yes,
carbon sink.
Carbon sink,
the wood density is very important. So the carbon sink issue, the volume growth is good and high wood density is good. That is a good for the carbon sink. So that point.
Okay, so wood density. Okay,
Density is very important.
Okay.
But in your country, sometimes the density increase with maybe the not cell wall component, like the extractives. Existence of this huge amount of the extractives makes the apparent density increase. So at that time the effect is limited. So the actual value of the cell wall, the density of the cell wall component, that is very important, I think. But sometimes I'm also confused that much. And sometimes the density, range of the density is the hardwood is very huge, but your country, almost the high density, you can find so many high density hardwood species. In that case, almost similar, high. So it's mean the volume is important. Growing characteristics is much better sometimes. But theoretically need the volume and density.
Okay.
I think so.
Thank you so much.
It's enough, Dr. Harry. Okay, thank you. Okay, for the last question, because it's almost break for Friday pray. In this room or in online? Professor Ganis?
Yeah, you mentioned destructive methods is sometimes not so good. It's not so good. It's about the method or the results. You mean?
Okay. Result is destructive is much better. Need the destructive test. If we conduct the destructive test, not only the destructive, non-destructive test usually get mainly the Young's models. The elasticity we can get the data, but some plasticity things like the strength properties, the actual how many load it can apply, that method cannot get by using the non-destructive test. So we should use the destructive test, but usually the people cannot allow to, for example, cut trees, and also need money. Yeah. And also my problem is not only the money, but money should budget. Okay, but so for example, the big size. The other one is chance. It's better to do the destructive test.
It's already juga yang Bapak Ibu adik-adik sekalian yang hadir secara online, apakah ada pertanyaan? Bila ada silakan. Well, ladies and gentlemen, that was the general lecture from Professor Futoshi Ishiguri. Please give a big applause for the presentation for today. Thank you very much. Hopefully it can be useful for all of us, especially adding our knowledge in aspect of the wood quality related to the tree breeding program. I represent the Forestry faculty. Would like to thank you to Professor Futoshi Ishiguri, who is pleased to be here to give a very interesting and useful lecture for all of us. Once again, I would like to thank you to the lecturers here, Professor Ganis, Dr. Hairi, Dr. Agus, Dr. Rizky, and others, and also for the master and also doctoral student who attend in this room or also in the online. Furthermore, I, as the moderator, would like to apologize if there are any mistake in hosting this lecture. It's all from me. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.